All right, here's our second video on enzymes. And what we just covered, just to look at this, in enzymes, we talked about activation and the energy barriers, getting over that hump, right? Active sites and the specificity, in other words, the key to the lock thing. Enzyme substrate complex, how it provides that nice environment for enzymes to do their thing. And now we're going to talk about the effects. How does temperature, pH, sorry that this indentation's off here. How does temperature, pH, concentration, and cofactors affect enzyme action? And then we'll eventually get to regulation, okay? So let's take a look at this, all right? And if you still have your diagrams, all right, keep these diagrams handy. We're on letter D, the effects on enzymes, and we're going to look at temperature and pH here. We're also going to talk about cofactors, and then we'll get into regulation, okay? So those are all the tools that you got to have with you on hand here. All right, so first let's talk about temperature. The temperature that an enzyme works best at is called the optimum temperature or optimal. You'll see I use those words back and forth. There's probably an English type out there that knows the difference, but not this science type. All right. Optimum is that perfect temperature, but this varies depending on the situation. Like in humans, our optimum temperature for our enzymes to work is anywhere between 35 and 40 degrees Celsius. That's body temperature. Kind of like, 98.6 degrees Fahrenheit. Okay. But an archaebacteria, that's those really weird bacteria that live in like hot springs. Their best temperature to function is 70 degrees Celsius. That's really hot. That's getting up close to boiling temperature. Okay. And an interesting thing about archaebacteria is actually the enzymes that are in archaebacteria that they use for a lot of genetic PCR, making more strands of DNA, which we're going to learn later in the year too. Okay, so the enzymes from these archaebacteria we can use at high temperatures, which are needed to make more DNA, make copies of DNA. All right, so a small increase in temperature can help, and that would increase an enzyme's activity. So you warm something up and it's good, but too hot will start to, there's that word we know, denature, change the shape of the enzyme, because remember the enzyme's a protein. Unless it's a ribozyme, then it's RNA, and that would even change the shape too. Heat would change the shape of a RNA too. And too cold obviously slows down. You know how you put ice on an injured part in your body or something, it actually slows down inflammation. Inflammation is another pathway that is, controlled by enzymes. And if you can slow down the enzyme action, you can slow down your pain. So that's why ice really, truly does work after a workout, especially on damaged tissues and whatnot. Okay, so that's how temperature affects enzymes. Little warmth, good. Too much warmth, bad. Little cold, slows it down. pH is another effect. pH, whether it's acidic or basic. Optimal or optimum, okay, is the perfect pH. With humans, we're mostly between 6 and 7, or 6 and 8, because remember, 7 is about neutral. So our pH is going to run in between there. But there is an exception. In your stomach, the pH, all the enzymes that work in your stomach, like pepsin and ghrelin, there's all these different enzymes that work in your stomach, have to work at a low pH. If it's not at that low pH, denature happens. So when the pH is not that optimum pH, you're going to have denaturing, whether it goes low or high. Now, I put this little thing over here, and I always think it's kind of interesting looking at digestion. And this is super important, you guys. And especially anybody thinking going into the medicine field or something. I love looking at, these are little approximate signs here. I love looking at the pH differences from the mouth, going through the digestive tract, the mouth, the esophagus, the stomach, the duodenum, which is the beginning of the small intestine, then the small intestine itself, then the large intestine, where you have the ascending, the transverse, and the descending to the colon, to the rectum, and on out your rear end. But what about the pH of these things? Let's start backwards, okay? So let's start your rear end, okay? Your large intestine is actually a 7.0 right by your anus, okay? Good thing, too, because you don't want to have a burny butt when you're pooping, okay? And this is why sometimes diarrhea has a higher acidic and it burns your butt, okay? But at the end of your large intestine, right before it comes out your rectum, your pH of your food is around 7.0, and that's what works best. any enzymes around that area. And you do have enzymes being secreted around that area. But in the beginning of the large intestine, you actually have a much lower pH of 5.5. So the beginning of your large intestine has a more acidic environment, which is interesting because the small intestine is very close to normal, okay, or neutral, I should say. It's very close to a 7. It's a 6.5, still on the acidic side. Remember, 7.0 is middle of the road. That's neutral. Anything below is acidic. Anything above is basic, all right? The duodenum, which is the beginning of the small intestine, right, that's at about a 6.0, a little more acidic, but it's a huge jump. When food leaves the stomach, there is a huge pH rise there that happens. Because in the stomach, the pH, and this is at the bottom of the stomach, even at the top of the stomach, it's not really a 2.0, but right at the bottom of the stomach before it leaves the stomach. And that's why you have like this sphincter muscle that once food leaves the stomach and goes into the small intestine, into this duodenum part, there has to be a sphincter because you don't want a backload, or else you get a lot of that acid indigestion that you hear about, right? When the acid goes into that section, a lot of times you will have a lot of problems in your duodenum, and that's where you start to feel that acid indigestion, okay? But your mouth is a 7.0, right? Except when you're eating spicy and hot food, right? Or if you're eating a lot of tomatoes, it actually is low. And the enzymes in your mouth, that saliva, that salivary amylase, work best at a 7.0. So, like if I was to draw the digestive system, let's see if I can draw it on the back here, like here's the mouth up here, goes down to the esophagus, right? Goes down into the stomach, right? And here's the duodenum, like in this section here. Then you go all into the small intestine. And then from the small intestine, eventually, you go up the large intestine, which goes up and down and around. I'll make it a 2. Here's your large intestine, goes up and around and down. And there's the rectum and the anus, right? So up here in the mouth, you got 7.0. Down here in the stomach, you're looking at a 2.0. In the duodenum, you're looking at that 6.0. All throughout here, you're looking at 6.5. But the large intestine is about a 5.5 here, and it ends up being about a 7.0 around here. So it's nice that in your mouth, it's kind of neutral. And at your butt, it's kind of neutral. But all in between, these enzymes will work at different optimal pHs. Okay, so I think that's always an interesting connection also to human and to being a doctor someday and talking about pH if somebody has acid indigestion. Now, you know why. It could be that little muscle from the stomach to the small intestine that's giving them problems. Or there's even another muscle. Let me bring this back out. That goes like right around here from your esophagus into your stomach. And sometimes you get that acid reflux. That's when acid from the stomach kind of goes backwards into the esophagus. The esophagus, that 7.0 is pretty much all the way down until you get to the stomach. And even in the upper realms of the stomach, it's not quite 2.0 until you get down to the bottom. Okay. So I really think that's interesting. I hope you found that interesting too. So that's pH. What else can affect enzymes? So temperature, pH, concentration. In other words, the more enzymes you have or the more substrate you have, the more action is going to happen. But remember, this is like a limiting reaction. If you're in chemistry, you remember limiting reactions. It's kind of like if you have so much enzymes and you only have a little bit of substrate, okay, well, that little bit of substrate is going to limit how much you're going to like deal with that substrate, okay? It's kind of like baking a cake. If you have like 5 cake mixes, but you only have 2 eggs, you're not going to be able to make 5 cakes. You're only going to be able to make 1 cake maybe with 2 eggs. I think actually they need 3 sometimes. But those are your limiting reactants. So concentration, more enzymes, more substrate. Yes, you're going to get more action, but it will only go so far because the enzymes have to work one at a time on the substrates. Okay? Cofactors. Now, cofactors are very interesting. These are like little helpers to enzymes. And so often people call them, they go right to calling them coenzymes, but they're cofactors. Things that help enzymes are called cofactors. Now, they can come in an inorganic form or an organic form. If they're inorganic, they're usually metals and ions. Okay, like calcium ions, sodium ions, metals such as like iron, Yeah, definitely like a metal. And those would be your inorganic cofactors. The organic ones are the true coenzymes. There's that word coenzyme. And again, you might learn in a first year bio that coenzymes are the helpers to enzymes because you think coenzymes. But really, coenzymes are only the organic or the biological ones that help enzymes. The inorganic ones are called cofactors. They're both called cofactors, but these in addition are coenzymes. Okay, enough about that. You got it. And finally, regulation. Okay, so now we talked about all the effects and we're getting into regulation. And regulation is going to come in a few things we're going to say here, but we're going to start off with the first, activation and inhibition. If you just look at those words, you know activation means you're activating something, you're getting something going. And inhibition, you're stopping it from going. Okay, activation means an enzyme is working, it's like working. Okay, inhibition means the enzyme has stopped. Okay, we got that down. Now, how do you activate an enzyme? By binding an activator to it sometimes. So sometimes the enzyme that's going to act like a key needs its own key to get it started. If that key is direct, okay, it will get into the active site, the regulatory site. It will kind of like, hey, let's open up our site and get ready to go. That would be a direct activator. But an indirect activator might attach somewhere else and say, yo, it's kind of like kicking you in the butt saying, get ready to get ready. Okay, what do I mean here? Let's take a look at this, okay? First off, I haven't looked at your diagrams in a while here. Oh, actually, I don't have the activator and never mind. Let's just put this aside for a moment. We'll get back to it, okay? Let's talk about inhibition. Inhibition is when an inhibitor gets into the enzyme and stops it from working. So an activator can get into the enzyme and start it up, and it can either directly get into that active site, that's like the key part, right? Or it can indirectly hit another spot, okay? If it's an inhibitor, it can be, and I don't know why they don't just call this direct and indirect also. Direct would be competitive. It's competing to like inhibit that active site. This is when it gets directly into the active site. That's why I think that they should call it direct. Non-competitive, I couldn't fill that all out, but you're going to see more of that there. It gets into what we call an allosteric site. So that word over here, allosteric, is somewhere else on the enzyme. Now it's time to look at that picture, okay? And we want to look at this down here. So here is the substrate in all these pictures, right? Here is the enzyme, okay? This here is the active site right here, right here, right here, okay? Now, this active site is perfect for the substrate, and sometimes that's normal binding. You do not need, anything to like start it, but you might need an activator in there that kind of smooths this over and gets it ready, or an activator that gets in here and gets it ready, okay, and gets it perfectly shaped. But here, so that's activation over here, and it doesn't show the two types, it just shows the regular type. For inhibition, though, if an inhibitor blocks that site, see, now that won't fit in there, right? That's called competitive inhibition. But if an inhibitor goes into another site, that's that allosteric site here. This is the allosteric site, and it changes the shape. See how this little thing went, and it's like doesn't have the perfect shape anymore? Then that's what we call non-competitive inhibition. Okay, so competitive gets right into the site. Non-competitive hits another site, an allosteric site. That's why we have the word allosteric here, and it stops it from working that way. Okay, back to the notes. All right, so where are we at in our notes now? We're almost done this video too, and we just started that. So we're going to look next at more inhibition, a little bit more and a few examples here. All right, so pay attention to the next video.